We will examine biosynthesis of tryptophan as another example of mechanism of action of enzymes. The actual details of biosynthesis of in living organisms, uh, tryptophan is synthesized from indole and serine. Indole replaces hydroxyl group of serine and is attached to carbon of the side chain of serine through a carbon-carbon bond. Now, there are problems with a reaction as written, because if we consider the way reaction is written, it would appear to be an SN2 reaction. And that in a way may make sense, because when we consider resonance structures of indole, of molecule of indole, we can see that indole has the localized free electron pair of nitrogen. That electron pair, due to the localization, makes carbon, carbon 3 of indole nucleophile. So that carbon now can donate free electron pair and act as a nucleophile to form a new, for example, carbon-carbon bond in this case. But problem is that for an SN2 reaction to occur, we need good or strong nucleophile and good leaving group. And in this case, actually carbon atom is a very poor nucleophile. Electron density is relatively low, so it's a poor nucleophile. And if you recall organic chemistry, you already know that hydroxyl group is very poor leaving group. So as written, this reaction is not going to occur. If you were to mix indole and serine in a test tube in suitable solvent, even with heating, this reaction would not occur, or if it were to occur, yield will be very low, extremely low. So to overcome this problem, we can catalyze reaction. So for example, we can apply base catalysis. Base catalysis, application of a base, would deprotonate nitrogen. That's the most acidic proton, one bonded to nitrogen. And now we have a strong nucleophile, because deprotonated nitrogen has 2 free electrons, and one of those 2 electrons is delocalized, and now that would be electron that carries negative charge with it, so carbon with free electron pair or partial free electron pair would also have partial negative charge, which means it would be much stronger nucleophile. So this nucleophile may be a nucleophile that is suitable for an SN2 reaction. But problem is that under the same conditions, basic conditions, serine would end up being also deprotonated. So side chain of serine would be deprotonated, and that would turn hydroxyl group into even worse leaving group, because now we would have an alkoxide, and that's actually not a leaving group. It's not that it's worse leaving group, it's simply not a leaving group. And so reaction could not occur under basic conditions, because simply nucleophile prepared from indole would have no leaving group to displace, because now serine does not have a leaving group. Well, we can attempt as an alternative acid catalysis. Acid catalysis would actually convert hydroxyl group of serine into a good leaving group. So hydroxyl group of serine would have been converted into oxonium ion, and that's a good leaving group, actually very good leaving group, and it could participate in SN2 reactions. But problem is that now, under the same conditions, acidic conditions, indole would be protonated. So nitrogen atom of indole would be protonated, and now indole would become completely non-nucleophilic, because there would be no free electron pair to donate. That free electron pair would have been used up in protonation reaction. Now there is no free electron pair to donate in a reaction, and so this species would be non-nucleophilic. So now we wouldn't have a nucleophile for an SN2 reaction. So neither acidic nor basic catalysis works. We actually need both at the same time, but in a test tube or a flask, that's not possible. We can't have both low and high pH at the same time. So a solution is an enzyme-catalyzed reaction. Enzyme for this reaction is tryptophan synthase. And tryptophan synthase is an alpha 2 beta 2 tetramer. So there are 2 alpha subunits excuse me, and 2 beta subunits. And starting material is actually not indole. It is indole 3-glycerol phosphate. It's shown here on the left. Indole itself is insoluble in water, and it's transported through cells as indole 3-glycerol phosphate. Indole 3-glycerol phosphate has large number of polar and charged atoms, and it's highly soluble in water. So starting material enters the dimer, the tryptophan synthase, in form of indole 3-glycerol phosphate. Now the enzyme tryptophan synthase utilizes pyridoxal phosphate, or PLP, as coenzyme. PLP is also known as vitamin B6. You will see that it is attached to the beta subunit of the enzyme as a Schiff base. If you recall organic chemistry, you will know what is a Schiff base. It's a compound that has carbon-nitrogen double bond. So there are 2 different types of subunits of tryptophan synthase. Alpha subunits convert indole 3-glycerol phosphate to indole. And then that indole travels through a channel in an enzyme to beta subunit where the actual reaction occurs. Reaction between indole, or reaction of indole and serine to give tryptophan. Here are some more details. So tryptophan synthase is written here is alpha 2 beta 2 tetramer. So alpha subunit, as already mentioned, catalyzes formation of indole from indole 3-glycerol phosphate, and beta subunit catalyzes the actual reaction of serine and indole. Alpha subunits in this ribbon model on the right below are shown in blue, beta in red, and you can see that indole 3-glycerol phosphate enters the alpha subunit, is converted to indole, and then it travels through a channel into beta subunit, where reaction is catalyzed by catalytic triad. So again, we have a catalytic triad, and in this case, it's composed of glutamate 109, serine 377, and lysine 87. And now we'll be covering the mechanism, so you will see the role of each. I should point out before I start covering the mechanism that we are actually not certain about all the details of the mechanism. So some of the steps are speculative, and I have indicated those speculative steps with a question mark. But they are actually pretty good guesses, as you will see. So here is the mechanism of biosynthesis of tryptophan. First step involves binding of the substrate, and it means binding of serine to the active site of the enzyme. And that binding involves exchange of lysine 87 side chain for molecule of serine. So molecule of serine enters the active site of the beta subunit of the tryptophan synthase. Lysine 87 PLP bond is broken, carbon-nitrogen double bond, and is replaced by carbon-nitrogen bond between PLP and molecule of serine. Mechanism for that is not shown here. If you recall organic chemistry, you can actually work it out. It's not very difficult. A number of additions and eliminations are involved. So that will be the first step, just binding of serine to the enzyme, to the active site of the enzyme. In the next step, alpha carbon of the serine is deprotonated. So hydrogen on the alpha carbon of this molecule of the serine bound to PLP is actually quite acidic and can be easily deprotonated because there are 2 electron withdrawing groups, carbonyl group of the carboxylate functionality and neighboring sp2 hybridized nitrogen. So amino group of lysine 87 side chain picks up alpha proton from alpha carbon of serine, and that results in formation of carbon-nitrogen double bond. That reaction is assisted by serine 377. You can see the cascade electron flow, how electron flow cascades towards pyridine chain of the PLP, and eventually pyridine chain of the PLP is, or not pyridine nitrogen, is protonated. Then the following step is elimination of water from serine. So a hydroxyl group of serine leaves as molecule of water. It's protonated by molecule of lysine. Note that, or not molecule of lysine, but side chain of lysine. Note that side chain of lysine 87 had just been protonated in the previous step, because in the previous step it picked up a proton from serine. Now that proton is used to form a bond to oxygen of the hydroxyl group of serine, and now hydroxyl group bonds to that proton and leaves as a molecule of water. Of course, water is a good leaving group. In the process, alpha-beta unsaturated carboxylic acid is formed. It's actually under physiological conditions in deprotonated form. So alpha-beta unsaturated carboxylate anion is formed. That's actually a very good Michael acceptor. It readily undergoes 1,4 additions. Again, if you recall organic chemistry, then you'll be familiar with that. So elimination of water from serine results in formation of very good Michael acceptor. And then in the next step, we have addition of indole. Molecule of indole is now added to this newly formed alpha-beta unsaturated carboxylate anion, and a reaction is assisted by glutamate 109. Glutamate 109 deprotonates indole to make it a stronger nucleophile, and now that nucleophile can readily undergo Michael addition to alpha-beta unsaturated carboxylate anion, and that forms carbon-carbon bond between what used to be indole and serine. And so now basically tryptophan molecule has already been synthesized by now. In the following step, deprotonation occurs to restore indole ring. A deprotonation is to basically restore aromatic ring. Deprotonation is assisted by serine 377, which basically takes back its proton. Note that in the step of elimination of water from serine, third step, that serine 377 donated, or sorry, in the second step, deprotonation of serine, serine 377 donated its proton to the pyridine chain of pyridine ring of PLP. Now serine takes back its proton, and that restores aromatic pyridine ring of the PLP. And in the final step, tryptophan is released, and now enzyme is ready for another catalytic cycle. And finally, we are going to consider the final step, step 6, release of tryptophan, in detail as an example of just how complex these enzyme-catalyzed steps are. And in fact, this is one of the simpler steps. So release of tryptophan from the enzyme involves the exchange of its bond to pyridoxal phosphate, or PLP, with lysine 87, with residue of lysine 87. It is actually a reversal of steps 1 and 2. And so in a reaction that is assisted by serine 377, and that's shown here by the arrow, and serine 377 is still deprotonated, so its side chain that is involved in this reaction is in the form of alkoxide. So in that reaction, aromatic pyridine ring of PLP is restored. The reaction occurs by alkoxide of serine picking up a proton from nitrogen. Then electron pair that used to be part of, or that used to be nitrogen-hydrogen bond, flows back to the ring, to the pyridine ring, to restore the aromatic ring. You can see at the top of the ring there is an exocyclic double bond. That double bond moves to form different carbon-nitrogen bond as indicated by the arrow, and carbon-nitrogen bond shown in blue flows, or its pi electron pair now flows to form carbon-hydrogen bond. So that becomes alpha hydrogen of tryptophan. So in this step, aromatic pyridine ring, so this is the hydrogen, that now becomes alpha hydrogen of tryptophan, and new bond is formed. And so in this step, aromatic pyridine ring of PLP is restored, and basically tryptophan is synthesized. But it is still attached to PLP. So in the following actually series of steps, there is exchange between residue of lysine 87 and tryptophan, where lysine 87 ends up being bonded to tryptophan, and lysine 87 exchanges with tryptophan for bond with PLP, and that releases tryptophan out of the enzyme cavity, enzyme active site, and restores the enzyme for the next catalytic cycle. So this is the bond, carbon-nitrogen double bond, to which lysine nitrogen adds, and in a series of addition and elimination reactions, exchange occurs. This is the amino group of lysine that participates in exchange. And here is lysine that leaves the molecule of the enzyme. So finally synthesized, sorry, not lysine, molecule of tryptophan that leaves the active site of the enzyme. And here is the same reaction sequence that we will write as organic chemistry reaction sequence. So if you are more comfortable, more familiar with organic chemistry, then this sequence may make a little bit more sense to you. And again, you can pause the video here and examine the sequence in more detail. And that completes our lecture on biosynthesis of tryptophan.